Thanks to everyone for standing by, and welcome to the Chimeric Therapeutics Investor Webinar today, discussing further detail from the chlorotoxin CAR T presentations provided at the Society for Neuro-Oncology annual meeting. All participants are in a listen-only mode. There'll be an initial presentation lasting for approximately 15 minutes, followed by some Q&A. If you have a question you'd like to submit, please feel free to type it in using the Q&A function within the Zoom app. Presenting on behalf of Chimeric Therapeutics today, we have the CEO, Jennifer Chow. I'll now hand it over to you, Jen. Go ahead.
Thanks so much, Matt. Whoa, is that better? There we go. Hopefully that's a bit better.
Okay.
Thank you, Matt, and thank you everybody for joining us this morning. We wanted to provide some insight into the further clinical data that was presented at the SNO meeting this past weekend. So just very quickly taking everybody back to where we were last week when the abstracts were released online, we saw the data as highly encouraging phase 1 data. We saw early signs of potential efficacy with a 75% disease control rate, where 3 out of the 4 patients that were treated at that first dose level actually had a best response of stable disease. We saw a very promising safety profile where there were no dose-limiting toxicities. The therapy was generally well tolerated, and so very promising there as well. And we saw encouraging bioactivity. We actually did see persistence of the chlorotoxin CAR T cells all the way through treatment, and we saw no signs of immunogenicity. So this is what was released during the abstract, online abstract release. When the data was presented this past weekend, we were able to get a little bit more insight into some of these different measures. So we thought that we would share those with you. So from an efficacy perspective, we learned 2 additional things this past weekend. The first is that disease control rate that was seen in 3 out of 4 patients lasted up to 8 weeks. And so that actually gave us our first sense of the durability of that disease control. The second thing that we saw, and I'll actually take you through it in a little bit more detail, was we're calling regional control of tumor recurrence, where the chlorotoxin CAR T cells were infused. And so this actually involves looking at the MRI scan of one of the patients. And what we were able to see is where the cells, the chlorotoxin cells, were actually infused into the patient's brain, there was no tumor recurrence. The tumor progression for the patient actually occurred in an area away from where the cells were infused. So that was actually highly promising to us as we move into dual routes of administration of this CAR. From the safety side, we did see the actual adverse events, and what we did note was that there was no cytokine release syndrome that was detected as well, which is obviously with CAR T therapy something that we're always watching out for. So there was no cytokine release syndrome. And then with Dr. Barish's presentation of his basic research, we actually were able to see in a little bit more depth some of the information that gives us confidence about chlorotoxin in very aggressive disease, and also some of the preclinical data will have us moving into new tumor sites, and melanoma is certainly one that's looking very promising from Dr. Barish's data. So I'm going to take you through this in a little bit more detail, and then certainly Matt will open up to any questions from that perspective. And these final posters with some of this information should both be available on the SNO website, and we'll look to put them on the Chimeric website as well today. So just the quick background again on chlorotoxin. Chlorotoxin was studied and designed for glioblastoma, which is a horrible type of cancer with a life expectancy of around a year. So really looking for new therapies. Chlorotoxin is unique in that its tumor targeting domain is a peptide derived from deathstalker scorpion venom, the chlorotoxin. And in the preclinical models, we saw very strong activity with chlorotoxin, even compared to other immunotherapy targets, which gave us really the resolve to move forward into the phase 1 trial we're going to talk about. So in terms of the trial design, the trial is designed across the 4 different dose levels, starting at dose level 1, which are the patients that we're talking about today, and it will move all the way through to dose level 4. Important to really note here, even for those of you that have probably seen this slide a couple times, because it's going to be important when we think about or look at the MRI scans, is how we're administering the therapy in dose level 1. What you see in dose level 1 is intratumoral administration only. And so essentially, there's a surgery, the patient has a catheter placed in the intratumoral cavity, and that's where we actually administer the cells. And at the dose level 1, we administer the low dose of 44 million cells. As we moved to dose level 2, which is currently the dose that's in progress, we actually added a second route of administration of the cells. So patients receive cells through the intratumoral cavity that we had previously, but now we've added a second catheter that allows interventricular administration of the cells. So now we're adding a second route of administration. So I'll just remind everybody as we start to look at the MRI scans that the patients that we're looking at only had cells through the one route of administration, the intratumoral route, and you'll see why that's so important. So what we saw in the abstract when it was released online was the 75% disease control rate, which really was very promising for us, considering this is the lowest dose of cells. Only 44 million cells were being given to a patient. And just a reminder that by the time we get to dose level 4, we'll be giving patients 10 times that amount of cells at 440 million cells. So 3 out of the 4 patients achieved an overall best response of stable disease by RANO criteria. And that is what was released last week. What you're seeing here is what's known, or what many of you may know, as a swim plot. So each of these lines represent an individual patient. So you see the patient unique ID down the side. What you see is when the patients receive the chlorotoxin cells, so that's marked in the black dots on each patient, and you see the response that the patient had. So what you see, if I use, for example, patient 487, 0 starts when the patient had their surgery. Then the patient had 3 administrations of the chlorotoxin CAR T cells, and they were assessed for response. When they were assessed for response, it was determined that they had a best response of stable disease, which is why the bar is then green. And then they were given 4 more chlorotoxin infusions before they went on to progress. And then you can see the survival that those patients had post-progression. So what you're seeing here across the 3 patients that actually achieved stable disease is that up to 8 weeks of disease control or stable disease was seen in these patients. 4 weeks, 6 weeks, and 8 weeks is what we actually saw. So that was an important new learning that we saw with the additional presentation of the data. To give a little bit of context to what you would expect to see, we don't have exact durability of responses, but we can look at progression-free survival. And so across different assets that are being developed for recurrent glioblastoma, so for the same disease, in phase 1 and phase 2 trials, so generally in phase 2 trials, it would be at full dose, and in phase 1 trials, it would be some patients at full dose and some patients at early dose. What you see is that unfortunately, glioblastoma, the median progression-free survival is still very low. So somewhere between 1 and 3 months is what we ideally would be looking at. So that up to 8 weeks, or between 4 and 8 weeks of durability that we're seeing at our very low dose, again, is for us very encouraging and certainly gives us enthusiasm moving into the higher doses. The next piece of information that was presented that wasn't necessarily in the abstract was MRI scans of patient 487. And so I'll take you through these because probably not everybody is necessarily used to looking at the MRI scans. What you see on the far left when you see preoperative is the scan of the patient's brain before surgery. The next column of scans shows you the operation, and so you can kind of see from the white piece here to here, the operation removing the tumor and where chlorotoxin was administered. So this was the injection site where chlorotoxin was administered through the one intratumoral cavity. What you see after 3 infusions is you see there's still no recurrence of tumor where the chlorotoxin cells were administered. But what you're starting to see, if you look at the last row here, is tumor recurrence happening where the cells were not administered. And again, when you look post 7 infusions on November 4th, this patient actually was determined now to have progressed. But what's noted is the progression did not occur where the chlorotoxin cells were given, up at the top here. It occurred where the chlorotoxin cells were not given. And so this was really important to us because what we're seeing is no recurrence in the left frontal lobe where the chlorotoxin CAR T cells were infused, but the recurrence and the progression happening very rapidly in the temporal lobe. And so this actually gives us really good reason for positive enthusiasm as we move forward. So where we saw no recurrence where the CAR T cells were infused and only recurrence where they weren't, in dose level 1, when we were giving single route administration, it gives us a great reason for optimism as we move to administering the cells in 2 different places now. So that ideally we would be giving cells here where there was no recurrence, and then also giving cells where in this particular patient there was a recurrence. So this regional control that was shown through this patient's MRI was very, very encouraging to us in terms of showing recurrence or progression only where the cells were not administered. From a safety perspective, this gives us a little bit more detail on some of the safety events. What we did see importantly was that there was no signs of cytokine release syndrome, and certainly with CAR T cell therapies, that's something we look for. Just highlighting on the right some of the things that we learned with the abstract release again, no dose-limiting toxicities, it was generally well tolerated, and there was a cerebral edema, a grade 3 cerebral edema event, but it was attributed at the possibly level, and certainly cerebral edema being something that's commonly recognized in glioblastoma patients. So promising safety is what we continue to see, or what we've seen even as we got into a little bit more depth in the data here. Dr. Barish actually presented at SNO a second abstract. So the first abstract was all about the clinical data that Dr. Brown presented, and Dr. Barish presented a second abstract with some additional basic research included. I've highlighted a little bit of it here. What you see on the left-hand side, where you start sort of having a little bit of green and some black in grade 1, and then a little bit more green in grade 2, and a lot more green by the time you get to grade 4, is these are the grades of disease. And essentially what you're seeing is MMP2 expression is what shows up as green. As you get to higher grades of disease, so more aggressive cancer, what you actually see is much higher expression of MMP2, which is what chlorotoxin is binding to and what is helping actually in terms of the binding and the killing of the cells. What you see on the right-hand side then, and again, these are, I'm sorry, difficult images probably to see, but what they're really showing you is the correlation between high MMP2 expression and the ability of chlorotoxin to kill. So the higher the MMP2 expression, the better you're seeing chlorotoxin CAR T cells able to kill. And so when you look at this together, what you're really looking at is high expression in advanced disease or a grade 4 disease, and high expression actually is killed better by chlorotoxin. So really suggesting that we could have benefit with chlorotoxin in the most aggressive disease. The other piece of important information that came out from Dr. Barish's presentation was early evidence for expansion into melanoma. So our plans are to open a phase 1 basket trial in other solid tumors in 2022. And this really starts to give us insight into which of those solid tumors are going to make a lot of sense to go into. What you see here is that chlorotoxin and MMP2 still have a very strong correlation in melanoma. And so this really is some early evidence that's going to give us that, or give us the information that we need to be able to determine that melanoma becomes one of those diseases that we look to expand into. And right now, it's certainly looking very positive with this early evidence that one of the diseases we'd be looking at that basket trial in is melanoma. So bringing it all together, the data that was released both in the abstract and then during the SNO presentation, very good signs of early potential efficacy between the disease control rate, the durability that we're seeing, and that regional tumor control, even through the single route of administration. Promising safety with no dose-limiting toxicities, no CRS, and a generally well-tolerated therapy. That encouraging bioactivity that we talked about when the abstract came out, where we saw persistence of the chlorotoxin CAR T cells and no signs of immunogenicity. And then the positive basic research insights that confirms that MMP2 is central to chlorotoxin recognition and killing, the support for moving or expanding into melanoma, and the evidence to support aggressive disease treatment. So highly encouraging data all around for chlorotoxin at SNO. Just reminding everybody, we're now in dose level 2 and progressing forward. And so we're giving cells both intratumorally and interventricularly at 88 million cells that are being dosed. And we'll continue to go to 220 and 440 million cells as we increase the dose levels. We're also, as many people are aware, as we move into the beginning of next year, looking to open up additional clinical trial sites for the glioblastoma trial so that we can recruit patients a little quicker as we move into those higher dose levels. And here you see the phase 1 clinical trial planned in other solid tumors, now with really some strong evidence leading us into melanoma first and foremost. Past that, so when we look at everything together, really advancing the phase 1 clinical trial with promising early data, moving our other asset that we didn't talk about, but the other asset, our CDH17 CAR T, making rapid progress on that development to be able to move that into a phase 1 trial in 2022, and continuing our work to round out our pipeline and our portfolio. So with that, Matt, I'll stop. I thank everybody for their time and hope that some of the insights that I've been able to provide you from the SNO presentation gives you a little bit more of a sense of what we've been seeing in those early patient cohorts.
Thank you, Jen. We'll move on to the Q&A session, as you've mentioned there. If anyone does have a question, again, please feel free to submit it using the Q&A panel on the Zoom app. The first question, Jen, is around the binding of chlorotoxin with glioblastoma cells. And the question is, does Chimeric need to discover how that works to be able, or resolve how that works in terms of seeking regulatory approvals?
No, so Matt, I think there are definitely some benefits from understanding the relationship between MMP2 and chlorotoxin better. But in terms of understanding, having a complete understanding to be able to file from a regulatory perspective, that is not necessarily required. We certainly are working on understanding, as you can see from Dr. Barish's basic research and his initial presentation, on understanding it though, because it becomes important for us to be able to assess whether MMP2 may be a good prognostic marker. So with patients, if they have higher levels of MMP2, will they respond better? Is there a threshold that below which patients don't have as strong as response? So it does become really important information for us, but it is not required for us to be able to file for a regulatory approval.
Thanks, Jen. The next question is, and I know there's probably a limited amount you can say on this, but what's the latest regarding the progress of Cohort 2?
Yeah, and so I think everybody knows that Cohort 2 is currently the dose level that patients are being dosed in. We anticipate that in the first half of next year, we will be dosing in dose level 3. So certainly we'll look forward to sharing the updates. As soon as we complete Cohort 2, we will let everybody know. We are very anxious as well to have that completed and move on to 3.
Thank you. The next question is, does MMP2 help tumour cells migrate, or what does MMP2 do or is responsible for in tumours?
Yeah, it's a great question. I think that what MMP2 does in terms of how chlorotoxin works with it is not yet fully understood. And I think that's one of the reasons why we are really invested in the research agreement and the research partnership we have with City of Hope to be able to further understand how MMP2 behaves and how it behaves in relation to chlorotoxin.
Thanks, Jen. The next question is also regarding MMP2, and it's what other solid tumours are known to overexpress MMP2 at a significant frequency, and how could chlorotoxin be delivered to them?
Yeah, it's a great question. That's actually something Dr. Barish is working on right now, is staining a whole host of other tumor types. What we do know from earlier literature is definitely melanoma was one that we expected to see in terms of it has high MMP2 expression. We also see MMP2 expression actually quite high in colorectal cancer. We see it high in some sarcomas, in head and neck cancer, in some breast cancers, as well as some small cell lung cancers. So across a number of cancers, we do see strong MMP2 expression.
Thanks again. The next question is around similar treatments and whether or not there's any possibility of early regulatory approvals based on the success of the trials.
Sorry, Matt, could you repeat the question?
It was just with regards to if there's any possibility of early regulatory approvals after these trials.
So from the phase 1 clinical trial, so right now we're in phase 1, what will be needed in terms of being able to move forward for a regulatory approval will be a phase 2 clinical trial after this. So normally in drug development, you sort of see a phase 1 clinical trial, then you see a phase 2 clinical trial, and a phase 3 clinical trial. We don't anticipate we will need the phase 3 clinical trial. We anticipate that our phase 2 trial, which would probably be a single arm trial in the neighborhood of 80 to 100 patients, will be all that will be required for registration. And with that, we actually believe, because of the high unmet need in glioblastoma, if the data looks strong, we'll be able to file for priority review as well. So certainly a relatively fast timeline to being able to move to a regulatory review or regulatory submission in cell therapy and glioblastoma.
Yeah, that's perfect. Thanks, Jen. The next question is, does Chimeric manage the data / see the data when it's conducting trials under its own IND? When we're opening the trial under our own IND, data is really cut at certain periods of time. So we don't look at the data every day. Really what it does is when you kind of finish a dose level, that's when you take a look at the data in its entirety. So under our own IND, that's what we would be doing, and we'd be managing the data access and availability then. Thanks, Jen. The next question is just around timing of the full phase 1 data readout. Do we have a rough timeline on when you think that will be completed?
For all 4 dose levels, Matt?
Yeah, that's correct.
Yeah, so if you look at it, essentially the trial was designed to be a 2-year trial, and it started sort of, I think, last September. So we would anticipate towards the end of 2022 is when we would have all 4 dose levels complete.
Great, thank you. Another question is, when will there be an update regarding the other asset that you've licensed and mentioned in the presentations?
The CDH17 CAR T. Yes. And so that CAR T, we are currently working on all of the evidence and all of the technical operations required to be able to move that into a phase 1 clinical trial. Right now, we've made some great progress. We've been able to confirm release of our plasmids, which is one of the very first steps we have to take to ensure technical operations are ready. And our plan right now is to have that asset in a phase 1 clinical trial in the second half of next year in 4 different tumor types: colorectal cancer, pancreatic cancer, gastric cancer, and neuroendocrine tumors. So that asset continues to move forward at a great pace, and we look forward to opening that trial next year.
Thanks, Jen. The next question is it necessary for the dose 2 cohort to be completed before moving to dose 3?
Yes. So in a dose escalation trial, what you really need to be able to show is not only that the dose level was completed, so once all of the patients in the dose level are dosed, you actually then have to wait 28 days to make sure that there are no adverse events or no serious adverse events, and file a report to, in this particular case, because the trial is going on in the United States, to the FDA, giving them a summary of what happened before you move on to the next dose level. So yes, dose level 2 needs to be fully completed, and patients have to be seen as having those dose limiting toxicities before we can move on to dose level 3, and then the same in dose level 3 before we can move on to 4.
Thanks, Jen. The next question is also back just on CDH17, and that is, are you planning a novel delivery approach as you have with chlorotoxin?
Yeah, it's a great question. With CDH17 at this time, we are looking at traditional systemic administration for the tumor types that we're going into. In terms of the gastrointestinal tumors, we believe those will respond better to the systemic administration with lymphodepletion. So we'd be looking to do a more traditional route of administration there.
Great. And another question that's come in is just with regards, you've mentioned in the past expanding the portfolio of Chimeric with other assets. Is that something that the company is still considering?
That is something that we're very actively still considering and pursuing, Matt, is we really are looking to ensure that we have a portfolio that is strategically designed and certainly rounded out. So the team is continuing all of the conversations to find those assets to bring into the portfolio.
Thanks, Jen. That concludes the Q&A. So I'll just hand it back to you if you'd like to just make a closing comment.
Yes, absolutely. So I just want to thank everybody again for their time this morning. And we will make sure that this data is available up on the website for anybody that wants to go through it in more detail. We're highly encouraged by what we've seen. It's very early days with just the first 4 patients at a very low dose level, but truly encouraged by what we've seen here. So look forward to providing more updates as the trial progresses.
Great, thanks very much for that, Jen. And that does conclude today's Chimeric Therapeutics Investor Webinar. Thanks to everyone for joining, and we'll see you next time.
Thank you.